Hi, welcome back to the podcast. For those of you who are new viewers, my name is Ashish. Today, we're going to be talking about something that has the potential to change the world, which is both controversial and revolutionary, making it a hot topic in the news at the moment. Scientists have successfully managed to grow mini chimpanzee brains.
So these brains are grown from stem cells.
Wait,
Um,
Kanishka, what are stem cells?
so stem cells are essentially the raw materials of the body. They're undifferentiated cells, and they can be forced into and coaxed into differentiating into other types of cells. So neural cells, for example, in this case, and then they can be forced to replicate in a lab situation, a
I
lab,
thought
lab
that
environment.
this
Sorry.
I thought that this was going on for a long time. So how come it's only coming back now? Because we've always used stem cells for certain types of treatment, right?
So yeah, well, stem cells are still relatively new, but they have been used in things like cancers, and they've been used to study different organs and stuff like that for a while now. But with the neural side of things, that's something that they've been trying to do for a long time. But new neurological differentiating systems have essentially enabled the actual growing of a new brain to occur. Um, these brains aren't really fully capable of any major functions like consciousness or anything yet, and they haven't even attempted to transfer consciousness because the brain isn't complex enough. But what these are right now is essentially neurons um, being amalgamated into these blobs in essence and kept in this gel matrix. And these blobs at the time kind of join together, forming a more complex structure when they just release sort of energy spark. Um, and those more complex structures are called cerebral organoids. But like I said, they're not actually capable of any huge functions. But they've tried to grow the brains of three different types of species. Well, three different types of brains in essence: human, chimpanzee, and macaque. It's interesting to note that they've discovered that the brains develop at the same speed originally, but once specialization to different neurons occurs, the macaque's brain develops the fastest, with the human brain being the slowest. And it's thought that this is due to the greater intricacy and complexity of our brains, according to the co-author of the study, Barbara Trutlin. But these studies have been really beneficial in aiding us in getting a better understanding of our own brain and the differences between our brain and chimp brain, despite DNA analysis showing that the DNA in cells is very, very similar. It does, of course, require more research, but yeah, it's really interesting.
I see. And what do you think the potential for this is? Do you think how far will it go, or is it more of just an experiment that they conducted to, as you said, help a study to identify the differences between a chimpanzee and human brain? I mean, it doesn't seem like it's going to be something that can be used for treatment in the future at this point of time. So, what do you think? What are your thoughts on that?
I would actually disagree. I think the potential is enormous. So, in the very basic state they were in a few years ago, they were still useful in understanding how our brains work, getting a deeper understanding of the neural capacity and understanding differences in the brain. Um, at the state they're coming to, we can really analyze key differences, and we can start to study complex diseases which were previously, kind of seen as very difficult and almost not impossible, but we had very little research on, in essence, such as Alzheimer's, how strokes affect the brain, stuff like that. They've recently started bleeding these brains when kept in the matrix, and this is a really significant step in treatment potential. So, if we look at stroke victims or other victims of degenerative and non-degenerative neurological conditions, the aim is to perhaps be able to replace brain structures with brains synthesized from the patient's own cells. So, um, basically, you can take a patient's cell from their, like during a neural procedure, for example, um, coax that into a stem cell and then recoax it into an endothelial cell, which line the blood vessels. So some work's been done on that with neurosurgeons to actually work with mice. And during a neural procedure, they took some cells. They took a few cells, coaxed them to stem cells, coaxed them into endothelial cells, and then fused those with the brains which were in the gel matrix. And then that fused brain was transplanted into a region of a mouse brain. And this mouse was actually alive and functioning within 2 weeks, 3 weeks of the cell, with capillaries growing in the brain, which is obviously really promising.
Yeah, that is pretty promising. I think that's probably the future. And I'm not sure about growing brains, but I think using certain cells and changing them slightly to benefit us is probably more realistic and a more approachable way of attending any neurodegenerative diseases,
I
because
mean,
obviously growing a brain is quite far off still. And I personally don't see a way that they can do this at the moment, because the max they've gone to is some brain-like organoids, which is kind of unpromising, if you know what I mean.
well,
It's not as exciting as someone would have hoped.
I'd argue that the fact that we're growing brains at all is very exciting. It poses a huge amount of issues and ethical issues, but um, the really interesting thing about it is that rejection would be prevented. So it's not like previous transplants of any kind, because it's a stem cell. If you take the stem cell from the individual, the patient, and then undifferentiate it, redifferentiate it, you're talking about a cell that's already been inside of the person, which means it's not going to have the same rejection capability that other cells do. Um, but that's kind of what makes it so promising. And studies done in other transplanted tissue so that blood capillaries are growing there, which is kind of showing the willingness and ability for transplantee capillaries to grow in the transplanted tissue. And that's
I
what is
see.
going to enable us to use it in transplant surgery potentially soon.
Okay. But I think anti-rejection is one part, and I think although that's a positive, there's also many limitations, which brings me nicely onto my next point. And
Yeah, I was going to ask actually, what are the limitations?
so at the moment, there is no way that to transfer consciousness. So scientists aren't even near measuring or even understanding consciousness. And I think this is the biggest obstacle we have. So if scientists can't even measure consciousness, right, not even understand what it actually is, where do we see a future where we can actually transfer it? I mean, sure, the brain shows some brain waves when electrical charge is applied across it, but essentially can't function by itself. And I think this frankly shows how far off this technology is. And I feel like it's better else to look elsewhere for more important and pressing issues, and maybe put this on the back burner a little bit till we get more advanced. And I mean, there's been a lot of experiments like that people couldn't explain during their time, but maybe the future generations can. So perhaps this is one of those where we just let it sit on the back burner and someone else takes it up in the future when we have better technology.
I mean, yes, I guess you could. I feel like doing that would also be kind of a waste, because you've come very far if you think about where we were with this, talking about literally not being able to use any of the technology we have right now a few years ago. In the short period of time, we've come to be able to grow a basic brain, and actually involve it in a transplant using mice. I feel like in a few years, yes, maybe not. I mean, you're talking about transfer of consciousness. That's only relevant if it's a whole brain you're talking about. If we're talking about just transferring a part of it to aid in stroke victims, for example, who the whole brain is not affected, I think you're looking at a really promising scenario here, and I think it kind of negates the issue of consciousness transference, because why would you need to transfer consciousness for a part of a brain, right?
Yeah, true, true. I agree with that point, and I think we should focus on the realistic goal of having certain cells being changed and differentiated. But I think there's also, I think, one other major issue is the ethical issue, and there's many religions against this sort of what they're trying to do. And I can speak like in Hinduism, the power of giving or taking life is believed to only lie with God, and hence this study raises countless objections. I mean, growing brains in a lab and then putting consciousness into it go against the basic moral principles. And also, in Hinduism, the soul is deemed to be the center of someone's life. So, how are we to decide when someone dies and they lose their soul to go and try and make them come back alive again by giving them a brain that's functioning again? So surely that would raise the objection of, are they really alive, etc. And it would be very confusing for everyone, for theists and scientists alike. So I'm
Yeah,
not sure if the ethical problems that this can have outweigh the help or the treatment that this could offer in the
I
future.
I think it's really based on what angle you're looking at. So personally, as a Christian, I can speak for the Christian faith. Um, Initially, when research was being done, embryonic stem cells were being used, and that obviously has its own ethical minefield because they're taken. Some are taken from aborted fetuses. Abortion is obviously frowned upon in most religions, but looking at this transfer of consciousness idea, you are again, playing God. I think you touched on the idea of like bringing people back to life. I think that's a very, very far way off. But I think if we were to even transfer consciousness on a small scale into perhaps like a vat-grown brain, um, that's not really something for us to be doing. I feel like from a religious perspective, I think it's almost us playing God. And some research was done into, when these brains. I think you mentioned how an electrical current was passed through. Um, they showed sensitivity to light, and scientists felt that they may be able to kind of feel the feeling of being trapped. And if we look at Genesis or Exodus 23:35, for example, you're seeing that inflicting mental or physical torment is wrong, as it is in any religion. So I think in that sense, it's definitely completely against religion. And you have to think about the fact that brains could technically develop a consciousness on their own naturally. And there are legal issues with that. Where's the consent if it's your stem cells that we used from one of your transplants? Do you own that brain? And
Yeah.
from a utilitarian point of view, um, I think they'd kind of sit on the other side of the fence. They'd say, do whatever's best for saving lives. And if you're thinking about the fact that 6.8 million people, I believe it is, die every year as a result of neurological disorders. And you're talking about saving, even if it's a sixth of those lives, I think a utilitarian would say it's a good thing to do. But I think currently I'm probably sitting on the side of it's a good up until a point. In transplantations and stuff like that, I think it's an amazing thing and we should definitely delve deeper. But in terms of transferring consciousness into a whole brain, I think it's probably a bit too far right now, and we really need to think about the ethical systems.
Yeah. I think so in summary, I think even like state of the art brain organoids, they lack the size, structure, and the connectivity between their neural networks to actually imitate a human brain. And they're also missing lots of important types of cells which we have in our brain. And as you said, they're starting to bleed, but they lack the blood vessels needed to keep their tissues like healthy. And I think overall, this just leads to the point that they're just immature slightly. And one technique for estimating their age has consistently likened them to the brain of a second trimester fetus. So they're obviously not yet being able to function on their own. And
Yeah.
I think it's been summarized best by Dr. Sung, who's a neuroscientist at the Paramount School of Medicine. He says that we are who we are because of our experience, and the organoid in a dish doesn't really have the stimulation to shape the brain circuitry in a way that it will develop more like a brain. So, I mean, in short, I think these organoids are nowhere near consciousness, and won't be for a very long time, I think, and maybe never even reach the point where we can make them into a brain.
I agree with that. And I think from my closing thoughts and my closing perspective, that they're an excellent idea in terms of transplant use for stroke victims, perhaps to, make potential full recovery, um, and for research purposes of studying diseases like Alzheimer's, etc. But I think we are a long way off transferring consciousness, and I think when that time comes, we should really kind of get ethical teams involved and really think carefully about what we could potentially do. But I guess that wraps up the podcast. I hope everyone enjoyed this week's episode. Make sure you join us next week for another amazing podcast. Make sure you like, subscribe, leave any comments you have in the Google Forms below. Anything you want us to discuss, leave it in that as well, or in the comment section. We'll be checking that. Yeah, follow us on Instagram. Thanks for listening.
Cheers, guys.